Welcome to Quick Course Digest. Today, we're taking a journey through the absolute essentials of the human biology lab. We're going to cover everything from how a scientist thinks to the incredible tools they use to unlock life's biggest secrets. You know, whether you're dreaming of being a scientist or you're just super curious about the world, the ideas we're about to explore are the real foundation of biological discovery. It all begins right here, in the lab.
Our journey doesn't start with a fancy piece of equipment, but with something way more powerful, a mindset. See, before you can do science, you have to learn how to think like a scientist.
So what even is science? Well, it's not some dusty old book full of facts. It's an active, ongoing process. It's a method for asking questions about the world around us and then finding answers through experiments that anyone can test and check for themselves. And that's the key right there. It's a shared journey to understanding. And to do that, you need a kind of mental toolkit. Critical thinking, that's your logic engine, helping you sort through information without bias. Skepticism is your quality control. It makes you ask, but is that really true? And maybe the most important tool of all is open-mindedness. It's the willingness to be wrong and to change your mind when the evidence points in a new direction. All right, with that mindset locked in, we can finally step into the physical lab. But hold on, before we even think about starting an experiment, there's one golden rule that trumps everything else. Safety always comes first.
And this is just so important to get. Lab safety isn't just about following rules to avoid getting hurt. It's about creating a stable, predictable environment where real discovery can actually happen. Think about it. Every single major breakthrough you can imagine was built on a foundation of safe, careful, repeatable work.
So what does that look like for you? Well, it starts with the basics. You got to protect yourself. So that means clothes that cover you up and shoes that cover your toes. Long hair, it gets tied back, out of the way of flames or chemicals. And the lab bench is a strict no food or drink zone. That keeps your experiments clean and it keeps you safe. Now, the lab also has its own universal language for danger. These little symbols, these pictograms, are understood all over the world. A scientist in Japan and a scientist in Brazil can look at that flame symbol and know instantly that something is flammable. It's a brilliant system designed for immediate life-saving understanding. And if something does go wrong, you need to know what to do without even thinking. This here is an eye wash station. If a chemical ever splashes in your eyes, you don't hesitate. You get here and you flush your eyes for at least 10 solid minutes. Seriously, finding this station should be the very first thing you do anytime you walk into a new lab. Okay, our minds are ready, our lab is safe. Now we get to the really fun part. Using the most iconic tools in biology to pull back the curtain on a hidden world that's all around us but completely invisible to our eyes.
You know, the microscopes we use today are amazing, but they stand on the shoulders of giants. It all started way back in 1665 with guys like Robert Hooke, who looked at a piece of cork and saw little boxes he called cells, and that name stuck. Then came Van Leeuwenhoek, who saw the first living microbes. But the real game changer was in 1830, when Joseph Jackson Lister perfected the modern compound microscope, really opening the floodgates for discovery.
All right, so you're peering through one of these incredible instruments, looking at something amazing. But how much bigger is it exactly? How do you figure out the total magnification? Turns out, the answer is some beautifully simple math. Your microscope has two main lenses. The ocular lens, that's the eyepiece you look through, and the objective lens, which is the one right above your sample. You just multiply the power of one by the power of the other, and boom, that's your total magnification. Now, these things are precision instruments, so you've got to treat them with respect. That means always carrying it with two hands, one on the arm, one under the base, for maximum stability. Always start and end on the lowest power so you don't accidentally smash a lens into your slide. And please, whatever you do, never use your shirt to clean the lens. Only use proper lens paper.
So we have the mindset, we have a safe lab, we have the tools. Now it's finally time to put it all into action. And we do that by following the time-tested recipe for discovery, the scientific method.
This is the fundamental roadmap that guides pretty much all scientific inquiry. It's this powerful, logical flow that takes you from just noticing something interesting all the way to a solid, evidence-based conclusion. You observe, you ask, you predict, you test, you analyze, and then you conclude. It's a beautiful process.
Okay, here's a really key idea. When you make a hypothesis, you're actually creating two. The first is called the null hypothesis. Think of this as the default, boring outcome. It's the idea that nothing will happen, that your experiment will have no effect. Then you have the alternative hypothesis, which is what you actually think will happen. The whole point of your experiment is to collect enough evidence to confidently reject that boring, null hypothesis.
So, how do you know your results are real and not just a random fluke? That's where the control group comes in. This is the group that gets the exact same treatment as everyone else, except for the one thing you're testing. It gives you a baseline for comparison. Without a control group, your data is basically meaningless. You'd have no way to prove your variable actually caused the change you saw.
The experiment's done, the data's in the notebook. But you're not finished, not even close, because a discovery is completely useless if it just stays locked away. You have to share it. And in science, that's what the lab report is for.
A good lab report is so much more than a diary of what you did. It's a scientific argument. It's your chance to prove that you don't just know what happened, but you understand why it happened and what it means in the bigger picture.
And that argument follows a very specific structure. You start with your hypothesis. You provide the background context. You describe your methods in so much detail that someone else could repeat your work exactly. You present your raw data, no interpretation yet. And then, in the discussion, you finally get to explain what it all means.
This is where your critical thinking skills really shine. You're like a detective, hunting for patterns in your data, trying to explain any weird outliers, and being totally honest about what could have gone wrong. And based on all that evidence, you make the final call. Was your hypothesis supported or not? And with that, the journey is complete. We went all the way from adopting a scientific mindset and staying safe, to using the tools, running the experiment, and finally sharing what we learned. That whole arc, right there, is the process of discovery in a nutshell.
So now you have the fundamental blueprint for asking questions and finding answers, the same process that's led to some of the greatest discoveries in history. The only real question left is, what mysteries are you going to uncover?